This is one of our first really good papers that show why COVID antiviral drugs are not prescribed for a long enough duration. With this paper focusing on Molnupiravir, which is one of the three antiviral drugs that are approved for COVID treatment, where Molnupiravir does accelerate viral load decline, but the virus is still detectable by day 5 in most cases, where only 14% of patients treated with Molnupiravir cleared the virus within the 5-day course of treatment, which becomes even more problematic because serial sequencing within patients revealed increased mutagenesis in the Molnupiravir treatment group, which means that the antiviral drug is putting significant pressure on the virus, causing it to mutate, meaning that the virus that the person is shedding or transmitting to others would then be a mutated version of what they were originally infected with. Therefore, the current 5-day treatment is too short, and longer courses should be tested to reduce this risk of potential transmission of Molnupiravir mutated variants being generated. So this study focused on non-hospitalized patients within 5 days of COVID symptoms that randomly received Molnupiravir, the 253 patients, or the usual care, 324 patients. This was part of a panoramic study, the platform adaptive trial of novel antivirals for early treatment of COVID-19 in the community, panoramic. And for primary outcomes, that initial study found that Molnupiravir did not reduce hospitalization or death, the primary endpoint. But for the secondary outcomes, it showed that receiving Molnupiravir experienced significantly reduced viral loads during treatment period and reported faster symptom recovery. Now, in this study, 94 patients agreed to do intensive sampling, and 563 opted in for less intensive sampling. And all participants provided dried blood spots on days 1, 5, and 14, where Molnupiravir was found to significantly increase the rate of viral decline during treatment, but once the treatment was stopped, so too was this rate of decline. And conversely, by day 14, viral loads were significantly higher in the Molnupiravir treatment group than in the usual care group, which is shown in this figure right here, where the Molnupiravir is the purple group, that's the antiviral, and then the control, the standard treatment, is the gray. So during those first 5 days of treatment, it rapidly decreases, it being the viral load. But once it's stopped, this rate of decline changes, and it actually, by day 14, is slightly higher than what you would get in the usual treatment group, which is likely a result of the mutagenesis that occurs on COVID from the treatment itself. So how long would you have to be treated, theoretically, to be cleared of the virus? Well, that's right over here. So using mathematical probability curves to extend this, if you are continuing treatment, then it would take approximately 10 days for about 95% of people to clear the virus on that treatment, which is what we've been saying for at least a year now. So where this study goes the extra mile is that they did genetic sequencing on the COVID itself from these patients. And they found that Molnupiravir-treated patients had a significant increased ratio of specific mutations, particularly Gs to As and Cs to Ts, which are those base pairs of our mRNA. And this was especially true in individuals that had virus persisting longer, so until day 14. They had consistently higher viral diversity compared to those clearing before day 14 and those without Molnupiravir treatment. Where this plot shows that, where from the start of treatment there's an increased number of mutations which continues on the treatment and then declines over time. But even with this generalized decline, if we compare that to the usual treatment, there's significantly more mutations or genetic diversity in the Molnupiravir group than there is in the usual treatment group, which provides a secondary emphasis on the need to extend the length of the treatment course so that you're not just encouraging a mutated version of the virus to exist. So here's that same data in another way. So the red is about 5 to 13 days, and the yellow is over 14 days. This is the usual care group, this is the Molnupiravir group. So the mutation load decreases over time in the usual care group, but increases over time with the Molnupiravir group, where even at day 5, we're seeing a pretty significant change, which is even more dramatic when you compare the groups at 14 days. Where the problem is sort of fourfold, but it all ties back into the same issue of the treatment is not long enough. So the first obvious issue is that this treatment promotes genetic diversity of COVID populations that you're infected with, so very rapid mutations. It's basically a positive or a negative selective pressure, depending if you're looking from the virus's view or the treatment's view, which ultimately forces the driving of new lineages of COVID. The second problem is because of this rapid mutation, it's taking longer for those patients to clear the virus because of the mutation, that transitional mutation that occurs. Because it's changing the virus just enough that your body's developed antibodies for the first thing it encountered. Now it's a little slower to start developing antibodies to the second or the third or the fourth thing that it's now encountered because of the quick changes. So there we have 3 points tied. We have an increase in genetic diversity, we have a slower time to final clearance, and we have a slower antibody responsiveness for final clearance due to the lateral mutational shift that's happening very rapidly, where all 3 of these things that I identified would easily be fixed with just having a longer dose, a 10-day treatment. And then the hidden thing, the fourth thing, is that we're giving positive selective pressure of the virus to mutate in such a way that it is resistant to Molnupiravir treatment. That by not having the appropriate treatment duration, that we're sort of also pushing it to become more resistant to the treatment itself. And that part's not covered in this paper, but that's also a fear of mine from what I'm seeing here.